In our last discussion, we looked at the origin of the universe, the origin of the Earth, and then three other major milestones in the history of life on Earth. And all of those milestones were accomplished by single-celled organisms. But at some point, we get more complex organisms. And there are two major jump-ups. One is when we first see early multicellular organisms, and the other one when we see more complex bilaterian organisms, which include all of the complex modern organisms today. And there's a dramatic difference between the complexity of mammals like us, hominids, birds, which are fairly complex also, reptiles, amphibians, and as we go down these multicellular organisms, arthropods have many fewer tissue types, cell types. By just about any measure, they're less complex than any vertebrate. And then there's simple organisms, which represent some of this early jump in diversity that are multicellular, but not nearly as complex as today's organisms. So realize there's a gradation of complexity across organisms, and mammals, birds, reptiles to some other reptiles to some extent are the most complex organisms. Okay, to get to multicellular organisms, however, we have to have eukaryotic cells. So major milestone number 6 are eukaryotic cells. And eukaryotic cells, of course, are more complex than single-celled prokaryotic cells, and there are no multicellular prokaryotes. So eukaryotic cells is among the hardest milestones to gather evidence for. We know it occurred. We know some of the things that had to occur to lead to a eukaryotic cell, but there's very, very little fossil evidence because, again, we're back at these single-cellular organisms. So the when is about 2 billion years ago, but this is less precise because we have very limited fossil evidence, but that's the best data we have to support it. So fossil evidence of the first eukaryotic cells, certainly by about 2 billion years, maybe a little bit farther back. That date is there's some uncertainty because of these gaps in the fossil evidence. Now, the eukaryotic cell makes up all of these complex organisms. Single-cellular eukaryotes are more complex than prokaryotes. But, of course, as we get more and more types of eukaryotic cells, we eventually have the possibility of multicellular organisms, including fungi, plants, animals, all of the major protozoan groups. And we know that the early eukaryotic cells were a fusion of a archaean and a bacteria. So the two major types of prokaryotes almost certainly fused together to create the first proto-eukaryote. In addition to that fusion, there is one other very critical, important step in the origin of the eukaryote, and that is gaining organelles. Now, the mitochondria and the chloroplast, some other organelles also, but these are the two big important ones that are present in most eukaryotes, all eukaryotes for the mitochondria, and in many eukaryotes, important eukaryotes for the chloroplast. But these were gained via something called endosymbiosis. Hopefully you've heard of that already. If not, let's review it very quickly. Many early single-cell organisms, and some still today, got their food through phagocytosis. So it essentially would engulf something on the outside, and by engulfing, basically they would think about pushing your finger into a balloon and then pinching off a little bit of that membrane out of the outside to engulf whatever you have brought in. And so notice that this, and this is important, this is a good line of evidence, notice that this outside element is now encased by a little balloon or a pouch of the original membrane that surrounds the organism that is engulfing it. So we now have this double membrane, the original membrane from whatever we engulfed, and then the outer membrane, which is a remnant of that phagocytosis process. And so mitochondria were originally a external living bacterium, probably something similar and closely related to our modern proteobacteria. But they were engulfed, and instead of being digested, they became part of the cell that engulfed them, and eventually became such an important part that the cell couldn't survive without them. So our mitochondria originated as external bacteria. The same thing happened in just a subset of eukaryotes, our modern green algae and plants, where they engulfed an ancestor of our modern cyanobacteria. And so plants can photosynthesize because they essentially gain the ability by engulfing a photosynthesizer, an ancient cyanobacterium. Okay, now the lines of evidence for this, there are actually three major ones and then some minor ones. But number one, the first line of evidence is the double membrane that we see around both mitochondria and chloroplasts. So that is a remnant of this phagocytosis event, this endosymbiotic, the beginning of the endosymbiotic relationship. Line of evidence number two is that both mitochondria and chloroplasts have their own separate genome that looks very similar in organization to bacteria genomes. It's more simple. It's been simplified. Genes have been lost. But its circular pattern, the way it's replicated, those are very similar to bacterial genomes. And so that separate genome for chloroplasts and mitochondria is good evidence that these were once free-living bacteria that have become now an integrated part of eukaryotic cells. The final line of evidence is phylogenetics. When we look at the genes in mitochondria and try to figure out those relationships of those genes, the mitochondrial genes are most closely related and their organization to proteobacteria, and the chloroplast genes are most similar to cyanobacteria. So this is why we know that these guys came from those ancient ancestors of those modern day bacteria. Okay, so we just know what endosymbiosis is. That's critical for the origin of the eukaryotic, and that we have a look at the mitochondria for all eukaryotes, and the chloroplast for those subset of photosynthesizing eukaryotes. Now, sometime after we have these proto-eukaryotic cells, right? These early eukaryotic cells, we begin to develop multicellularity. Multicellularity most likely originated as a transition from colonial, which is just single-celled organisms that are clustered together. And we transition to cell specialization. And what defines the individual is this specialization of cells, and particularly a separation of the germ line, right, which are the reproducing cells, and the somatic line. In single-celled organisms, there is no germ line, somatic line, it's just cell line, and everything can make copies of itself. But at some point, we get this differentiation. Now, there are many, many advantages. These are emergent properties as we move from being single-celled organisms to multicellular organisms. These are just some of them. You don't need to study them in depth or detail, but realize that there are many advantages that multicellular organisms have over single-cellular organisms. There are a few disadvantages also, but these advantages far outweigh the disadvantages. And there are two major groups of organisms that we study when we are studying the origin of multicellular life. These are living organisms, and so they serve as good models for the origin of multicellularity, but for different reasons. The Volvocene algae we study because there are many closely related species. Some are completely unicellular, some are colonial, so they group together, but individual cells can still all reproduce. And then some of them are simple but truly multicellular organisms. So these organisms have preserved in different species some of the steps along the way to multicellularity. In the slime molds, it's a little different. Now, slime molds are not part of the fungi kingdom, despite the name mold. They're more closely related to amoebas. But these slime molds have, in a single species, different life stages that are single cellular, that are truly little amoebas crawling around on the floor, the forest floor. And then other stages where all those single-celled organisms group together and make a kind of colony, but then in that colony, some cells specialize and put up these little reproductive structures. So only some of the cells reproduce. So these slime molds are kind of caught right at that transition point, and they do well that way, and they may have evolved it fairly recently, or it might be an ancient thing that they just have been able to utilize, where they can do kind of get the best of both unicellular and multicellular worlds. Now, the date for the origin of multicellularity is also a little tricky. There is a little bit more fossil evidence than the date for the first eukaryotic cells, but the other reason it's tricky is something different. All of these events we've been talking about so far evolved a single time, and then were organisms that were moving forward, but all of the evolutionary events were unique. But multicellularity, as we look at it across life on Earth, certainly has evolved multiple times. So this is a phylogeny here, a diagram showing relationships in major groups. The ones in red are the multicellular groups. And notice we have at least 1, 2, 3, 4 major groups, right? So we've got the plants and then the red algae. We've got these slime molds we talked about, some of them which are truly only multicellular. And then we have animals, and of course fungi, which have many. So the yellow ones have some multicellular groups and some single cellular. So several times multicellularity has evolved. The earliest was probably about 1.2 billion years ago, but other multicellular evolutions may have occurred much more recently. Finally, the last major milestone that we are going to cover for this run-through of these things is the origin of complex organisms. And by complex, I mean bilateral with multiple cell types, not a sponge, not a jellyfish, but these more complex organisms with brains, with sensory organisms. And this occurred about 540 million years ago. The Cambrian Explosion is what it's called. And it's just simply when we see a dramatic, rapid origin in the fossil record of these complex organisms. We see all major phyla. We see vertebrates, or chordates really technically, but eventually vertebrates. We see arthropods, we see annelids, we see mollusks. We see all of these evolving and appearing in the fossil record for the very first time. And we're going to highlight some of those organisms in the next group. Now, I put this but here because although we covered 8 major milestones of life, there are many others. And in fact, people might make a different list than I have done. But you should know those 8, know the dates, and know the supporting data for those dates. For many of them, it's just the relative dating of fossil evidence. But we could also look at the origin of sexual reproduction, which we'll do in a later chapter. Some of these we do in later chapters a little bit. We could look at the origin of movement, the origin of sight, origin of consciousness. All these we could make a case for being major innovations and important milestones in the history of evolution. If you're interested in them, you can look up this book. It's not required reading. It's purely if you're interested. It's well written. It's for kind of an intermediate lay, scientific versus novice audience. So it's very accessible. If you're interested, read it, and it will go over more of these in more detail. Some of them we'll talk about in more detail later. But for now, know the dates of those major 8 events. Now, before we finish up this lecture, I want to talk about the fossil record. A fossil is simply a preserved remnant of a living organism. It can be a piece of the organism. It can be a track of that organism walking over a mud flat, for instance. It could be droppings, right? So anything that gives us some information about an ancient living organism. And the vast majority of fossils have none of the original material, right? So Jurassic Park, I've put this insect preserved in amber here. The main conceit, or the beginning point of Jurassic Park, was that they were able to recover preserved DNA from amber fossils. In reality, there's really very little or no DNA left, even in fairly recent fossils. And in most mineralized fossils, what we typically think of as a standard fossil preserved in sedimentary rock, there is none of the original carbon, nitrogen, oxygen, and hydrogen that made up that organism. It's all been replaced with minerals. So although we can see structure, sometimes even very fine structure, none of the original organic material is left. But it still can tell us a lot about that organism. Now, to understand fossils, we need to understand 3 types of rocks. We have igneous rocks, which are formed from molten lava, magma. Okay? We have sedimentary rocks, which are formed from bits and pieces of many different types of other material. They can be from living organisms, which is how we get fossils. They can be from sedimentary rock, which has eroded and weathered, or other types of rocks, igneous rock that has eroded and weathered. And all those together create sedimentary rocks. And they are created at the bottom, sometimes in vast distances, but they're created at the bottom of bodies of water like oceans or lakes or streams. And so as that sediment, as those organisms all accumulate at the bottom and then get compacted and forced under pressure, we form sedimentary rocks. And then finally, we have metamorphic. These are the least important for biology. Metamorphic rocks are rocks that begin either as igneous or sedimentary and then are changed dramatically through pressure or heat, or both. And that's a metamorphic rock. So it starts as one of the other two and then is changed by pressure and heat. And marble is a type of a sedimentary rock, or a metamorphic rock. Sometimes they're quite beautiful, but often have very little scientific evidence when we're looking at fossil evidence. Now, I'm going to do a direct dating exercise, and I'm going to show you how we can directly date igneous rocks, because there's a little bit of this of a catch-22. Igneous rocks we can directly date using this method that I'll show you here in just a bit. Sedimentary rocks we cannot directly date. And the reason we can't directly date sedimentary rocks is because they are an amalgamation, a mishmash of all different things that originated at different times. So in a way, dating a sedimentary rock would be like trying to date a class, for instance, of students based on their ages. So if I go into a college class and say, how old are you? You're 18, you're 21, and then I say, oh, look, this class is about 19 years old because of the average date of its students. That's not accurate. We're dating the individual components, not the class itself. And so if we try to date the components of a sedimentary rock, it's very, very difficult, and it doesn't give us an origin for when the rock formed. Not only that, but many of the components that are fossils no longer have the original material there, so we can't directly date them. So this is the catch-22 of fossil dating. We can date igneous rocks accurately, but there are no fossils in them because they are basically just cooled molten lava, and fossils that fall into molten lava would immediately get burned up. We can see good evidence of living organisms, and we'd like to date sedimentary rocks, but we can't directly do that. So how do we get around this process? Okay, so let's talk about direct dating of fossils first. And to do this, I'm going to bring up a Word document. Let's bring up a brand new one. Okay, and I'm going to type here a little bit so you can see how we would do this. So, direct dating begins, there are three things that we need. Number one, we need to have an estimate of original radioactivity. Now, luckily for us, we can study the formation of igneous rocks today. We can study ones that we know formed at a certain period based on historical evidence, and we can estimate the original radioactivity. That's not very hard. Number two, we need to have a sample of a similar rock with unknown age. So for instance, if we know that certain types of granite have a beginning level of radioactivity, and we find that type of granite, we now have a beginning estimate of radioactivity, and we can measure a sample of a similar rock with unknown age, and then we just have to measure its radioactivity. And then the last thing that we have to know is we have to have a known half-life. And that's something that's a physical characteristic of radioactivity, and we can do. So let me give you an example. Here's the exercise. We'll do a couple of these just so you can practice. Okay? So let's say we find an ancient rock with a radioactive level of, let's say, 3 picograms of whatever radioactive level we're measuring. Now, typically we use uranium because it's got a very long half-life, and it's an element that's common enough in many igneous rocks that it's useful. But whatever radioactive level. So we find an ancient rock with 3 picograms of, we'll say, uranium. Okay? And now I recognize that type of rock. I recognize that when that rock is formed, it has 48 picograms of uranium, that same amount. Maybe we're looking at a kilogram of rock or something. And in that same amount of rock, we have 48 picograms of uranium. And we can measure this by measuring the radioactivity of that rock. Okay? So the question is, how long does it take us to go from 48 picograms, which is in that original rock, down to 3 picograms? And to calculate this, you need to use logarithms, but we're not going to do that for this class. It's a little complex. It's not too bad if you've got a scientific calculator, but I just want you to understand the concept and be able to do an approximation of this. So let's think about this. If we start at 48 picograms, how long does it take us to go all the way down? We'll put it way over here. How long does it take us to get to 3 picograms? Now, to do this, we have to understand the principle of half-life, which is just a physical property of radioactive materials. So to go from 48 to half of that material takes 1 half-life. So to go from 48 to 24 would be 1 half-life. So we'll put a little arrow there to represent that half-life. If we were then to go down half again to 12, that would take another half-life. And this is just the way it works. Why doesn't it go from 48 to 24 to 0? Well, that's not the way radioactivity works. And we could get into the physics of that a little bit, but you just need to take it as a property of radioactive material. The next major jump is down to 6. That's another half-life. And then finally, when we divide 6 in half, our final jump to this half-life, let's erase these, goes down to 3 picograms. So we go from 6 to 3 picograms. So if you draw a little diagram like this, put your original amount of radioactivity, have your final target, and figure out how many half-lives it takes. So we have to count 1, 2, 3, 4 half-lives to go from 48 picograms to 3 picograms. So if the half-life of whatever radioactivity we're looking at, it's actually much longer than this for uranium, but let's just for simplicity say the half-life of our radioactive element is 50 million years. Okay? If that's true, then all we have to do is say, oh look, 4 half-lives to go from our ancient sample to our the modern sample, right? The one that's forming today has 48 to our ancient ones. It takes 4 half-lives. Therefore, all we do is 50 times 4. And so our sample is 200 million years old. Okay, so that is how we directly date radioactive rocks. Now, remember, this is for igneous rocks that have radioactivity. So we can date igneous rocks, but there are no fossils. So we still, even though we can directly date these igneous rocks, we still need to somehow get around this catch-22 of not being able to directly date sedimentary rocks. So let's do one more. I'll let you practice it. I'll give you a little bit of time and then the answer here. So let's say we're going to start with a sample that has 128 grams or picograms, right? The small amount of radioactivity. So when rocks are first formed, they're 128 picograms. An ancient sample only has 4 picograms. So if the half-life of this radioactive element is, let's say, well, we'll just say 50 million years again. Let's say it's the same element. It's got a half-life of 50 million years. Okay? Your job now, and you should be able to do this, is how old is the ancient sample? Okay? And you can do the same method. So pause your video here and try and figure it out on your own. Start with 128. Draw an arrow representing a half-life. Go to half of that, half of that, half of that until we get exactly to, or at least close enough to 4 picograms. How old is our sample? Pause the video and try and do that on your own. Okay, here's how it works. 128, right? We're going to take 1 half-life and get to 64. We're going to take the next half-life. That would take us to 32. A third half-life is going to take us to 16. Not really complex math here. Now, if you'd like a calculator to do this, that's fine. I'm okay with that. But you need to understand the concept of how to do it. 8 would be the amount after our next half-life. And then finally, after a 5th half-life here, we would get to 4 picograms. So to get from our original sample down to 4, it takes 1, 2, 3, 4, 5 half-lives. And so all we would do is simply say 5 half-lives times, right, we have 50 million years per half-life. And that, of course, is going to equal 250 million years. So we can directly date many igneous rocks this way, and uranium has a long enough half-life that we can span the entire history of the Earth if we know a beginning amount of radioactivity. But we still haven't got around the catch-22 of how do we date fossils, which we can't do this directly to. If we try to directly date a fossil, a sedimentary layer that has fossils in it, we're going to get some random nonsense number because we're, again, just estimating the elements that went into the rock. We don't know when the rock itself formed. Okay? So let's talk about how we get around that catch-22. To do that, we use so we find fossils only in sedimentary rock and metamorphic rock, and really only the very useful ones in sedimentary rock. And we don't find any fossils in igneous rock. So to get around this, we use two elements. We use the direct dates that we can get from igneous rocks, and then we use a concept called stratigraphy. And this is a rather easy concept. If we have sedimentation occurring, so here we have dirt material all accumulating, a dead organism in it, maybe an ocean or shallow lake over it, and then continued sedimentation. When we look at these layers of rocks, and you can see these in geological deposits, right? Drive up to San Antonio and you see all the beautiful layers of white limestone, some a little different color. You can see these banding patterns. When we look at these strata, that's what the layers are called, and this is why it's called stratigraphy. The layers on the bottom are the oldest, right? That's pretty obvious. You can't put a layer on top of something unless what is underneath it is formed first. So we have oldest layer, next oldest, all the way up to the top, which are the youngest layers. So that and the direct dating of igneous rocks allows us to get a relative date for fossils. And so we use stratigraphy to escape the catch-22 of fossil dating. Now just very quickly, I just want to review this. Fossil dates are often not incredibly precise, but they are accurate. And so you need to understand those concepts of precision and accuracy in general, and of course especially as they apply to the fossil layers, these rock layers, and then the fossils we find in them. So here's what I mean. Precision is how sure am I that it's exactly that number. So for instance, if I ask you to, or if you ask me to guess your age, and let's say you say, I'll give you $100 if you guess my age. And I say, okay, done. But we don't come up with any parameters about precision. I say, okay, you are somewhere between 5 and 105. Now, is that accurate? Yeah, I got your age somewhere in that span, but it's not very precise. Ideally, we have both precision and accuracy. But if we have to give up one, we give up precision. And we don't always have to give up a lot of it. So when you see a fossil date, for instance, you'll see this fossil is between 45 and 48 million years ago. So that gives you an estimate of precision. And if we're careful about the way we do the work, that can be very, very accurate. So just be aware that fossil dates are always, if we have good methods, are accurate, but are not often very precise. But that's okay. So let's look at how we finally escape this catch-22 using the direct dates of igneous rocks and then the relative dating that we have in stratigraphy. So if I have both sedimentary layers, representing here by these horizontal layers with different patterns, and then I have different igneous events, I can get a relative date for these Eocene rocks by looking at the dates that we're able to gain and get directly from igneous rocks. So for instance, here we have an igneous lava flow that comes up through sedimentary layers, but then it doesn't interrupt as the Oligocene rocks. So we know that this lava flow is younger than these three sedimentary layers. Right? Hopefully that makes sense to you. If it's going to flow through it, it has to have occurred after, so it has to be younger than these sedimentary layers. But it's older than the Oligocene layer because it does not go up and interrupt that Oligocene layer. So now I have a window of time for these Eocene sedimentary rocks. I know that they are younger than 57 million years, right? So they occurred somewhere because they're laid down on top of this lava event that's 57 million years old. I know that they are older than 36 million years. So now I have a window. So I would date the Eocene sedimentary rocks at between 36 and 57 million years ago. And the Oligocene rocks, I know because they're laid down on top of this lava flow. And this other one I could date between 20 and 30 million years ago. The Paleocene rocks here, I would know are older than 57 million years old, but that would be the best I could do. So it's not a very precise estimate because I would say 57 million to 4.5 billion years. But if I had another lava flow, maybe even hundreds of miles away, that interrupted these same Paleocene sedimentary rocks, I could get a bit of a more narrow window. And so this is how we date fossils. We do it very accurately, but sometimes not very precisely with some windows of estimate. And so I would look for a lava flow that I could date, maybe even far away, but in the same type of rock, the same layer of rock. And by doing this, getting direct dates for those fossil layers and then putting in their proper stratigraphic location, I can get very accurate dates for fossils. So we've gotten around this catch-22 of fossil dating by using direct dates of sedimentary rocks, direct dates of igneous rock, and then helping us those to establish the relative date based on the layers of those sedimentary rocks. So on the worksheet number 1, you have a problem that helps you review this. But if you are given a diagram like this, you should, without any direct dates, be able to give me some relative dates for the different sedimentary layers. Paleocene rocks, the best you can do with this diagram is more than 57 million years ago is when they formed. The rocks beneath the Paleocene rocks, we know they were more ancient, but we wouldn't have any good, even less precise. So we'd say older than the Paleocene rocks, which are older than 57 million years old, but we don't know how much older. So we need to gather additional data if we wanted a more precise date. Okay, so look at the worksheet, review that. Given a diagram like this, you should be able to give me relative dates of sedimentary layers. You should also recognize that if you tried to directly date by taking a radioactive measure of a sedimentary rock, you would not get any reliable information out of it. And that's part of the worksheet too. So in the worksheet, a assistant who just doesn't know sends off all the rocks to be dated. And in reality, that would never happen. But in this hypothetical example, it does. And so you need to recognize that any direct date you get back from a sedimentary layer, you cannot trust. The only direct dates you can trust are from these, sorry, I had a little brain fade there, these igneous rocks. Those are the only direct dates that you can trust.